So it's exciting to talk about this work just presented by Greg Riley about a new drug, TAK-788, in non-small cell lung cancer. So it's for a very, very particular type of non-small cell lung cancer, EGFR mutant lung cancer with exon 20 insertion mutation. So only 5% of EGFR mutant lung cancer overall, and EGFR mutant lung cancer is only about a 15% slice of the non-small cell lung cancer pie overall. This was a phase 1 and phase 2 clinical trial. So phase 1 trying to find the right dose of the drug, and there were a number of different expansion cohorts, over 10 different expansion cohorts, ultimately settling on 160 milligrams every day as the maximally tolerated dose. Then that dose was selected and moved into a number of extension cohorts, and these data are mainly presenting the data from one of those extension cohorts, a total of 28 patients who received the drug. Out of those 28 patients, the responses seen, radiographic responses of tumor, were seen in 43% of patients. And the progression-free survival time was 7.3 months. And this compares favorably to other drugs in its class, and much, much better than we expected from other EGFR inhibitors out there.
And was there an association between TAK-788, the response of it, and particular EGFR insertions?
So within the EGFR exon 20 insertion mutations, we did look at the subtype, although clinically we tend to categorize all of these together, and it didn't look like there was any clear association of any of subtype mutations having a response.
And how does the efficacy of this drug compare with other EGFR tyrosine kinase inhibitors?
So what's unique about EGFR exon 20 insertion non-small cell lung cancer is that the FDA-approved EGFR tyrosine kinase inhibitors have been tested, but are minimally to not effective in this type of non-small cell lung cancer. What's also interesting is there are other drugs tested in its class, and one of them, poziotinib, has some data presented. However, these data look a little bit stronger overall in terms of the numbers that we've seen overall. Plasma biomarkers were not tested in this study, although certainly plasma biomarkers have been developed as a way to detect this type of non-small cell lung cancer overall, and could theoretically be used to follow responses over time. So overall, these are promising data. This is showing responses with a novel drug that's potent and selective for a novel target. Right now, there's no FDA-approved drug that seems to substitute for the effect that we've seen here. So what's going to happen is this drug is being moved into an extension cohort with many more patients being enrolled, and the results of that will hopefully help validate this. So overall, this drug looked like it had toxicity very similar to what we see from other EGFR inhibitors, diarrhea, some nausea, vomiting, some rash, as well as an unusual toxicity, which was pancreatitis, but mostly chemical pancreatitis, not clinically significant. So it looks like a tolerable drug overall.